Well, hello everyone. My name is Coralie Bouillot. I am a member of the board of Sjögren Europe, and I'm very pleased to welcome you to this webinar, which almost ends Sjögren Awareness Month. We hope you have been able during this month to hear from our members, their actions and challenges. Thanks to them who sent their videos and testimonies. We really think that this will help others to understand better Sjögren disease and will contribute to raise awareness. Of course, as you can imagine, all of them expressed the same wish to see the emergence of new treatments for Sjögren soon. So where do we stand today? What are the research directions and prospects for patients? To answer those questions, we have the great honor today to welcome Professor Xavier Mariette, Head of Rheumatology in Kremlin-Bicêtre Hospital in Paris, in France. Professor Xavier Mariette is internationally known for his researches in rheumatology, especially in Sjögren's, and he is very involved in patient-centered medicine and values patient feedback. So on behalf of the Sjögren Europe board, I would like to thank very warmly Professor Mariette for accepting our invitation and more generally for his commitment to Sjögren patients. After his presentation, there will be a question and answer sessions. If you have questions, please write it in the dedicated question box. The chat box is only for comments. And please note that this webinar will be recorded for dissemination purpose. So Xavier, thanks again. And now the floor is yours.
Thank you so much, Coralie. Thank you to all people from Sjögren Europe. It's a great honor and a great pleasure for me to be with you tonight, at least tonight in Europe, but I saw people came from US, maybe not so many Asian people because of the time, but it will be next time. So firstly, I want to congratulate you for the initiative you took in this organization, because it was absolutely mandatory that all patients all over the world could organize themselves in some strong organization. And you do know that the Sjögren Foundation in the US met this objective, and really I congratulate you for having succeeded, for having shared together the strengths and the forces for better collaborating with the clinician and the researchers, and for better talking together about your disease, because it is your disease and it is because of your symptoms and because of that we have to continue to search and to improve the knowledge and the treatment of this disease. So today I am going to tell you about the new the progresses which have been made in the disease. So it's very difficult to choose what are the latest news, the most important progresses. You can see my slide, it's okay, Coralie?
Yes, perfectly, okay. Thank you.
So it is the plan I decided to follow. So do not hesitate to ask questions in the chat, or we will have time for the question after the talk. I will speak 20-25 minutes and we'll have time for question later. So I would like to emphasize 3 points. The first one is that the mechanism of disease is probably different from one patient to another. So it is crucial today to stratify the patients, because the treatment will not be the same for patient A and patient B, probably, because there are subgroups of patients. Because it is the most important for you, I would like to give a message of hope, because we now have new promising drugs with the first clinical trials which are positive in the disease. And that's very important for the patients. And lastly, we will finish with the NeceSSity project I am coordinating, just to give you an example of the flavor of what could be a big research project with different countries, different partners, and the importance of the involvement of the patients in this NeceSSity project. So let's begin by the necessity of stratifying the patients. I show you this slide. It is just to show you that the mechanism of the disease involves numbers of actors. numbers of cells are involved for explaining this disease, which is an autoimmune disease, explaining how autoimmunity can occur to finally make injury to the glands, to other organs, and to the symptoms. You see, the epithelial cells in the glands, salivary glands and lacrimal glands, and you have these cells which are called PDC, which are these cells are secreting a cytokine which is called type 1 interferon alpha. And you have also, of course, the immune cells, but all the immune cells are involved in this disease. The T lymphocytes, the B lymphocytes, the plasma cells. So it's difficult finally to see in one patient what is the cells, what is the pathway, what is the mechanism which is the most important. So it is necessary to do what we call a stratification. And I show you this recent publication from my colleagues in Brest in France, Jacques-Olivier Perse, and they could make stratification of the patients just by a blood sample. Taking a blood sample, they look at the expression of genes in the lymphocytes of the blood. So the lymphocytes are the blood cells, the white blood cells, which are useful for defending the body against infections. And you do know that when you have autoimmunity, these lymphocytes may secrete autoantibodies and may attack the self, and it can lead to autoimmunity. You can see there are 4 different subgroups in this analysis. It means that these patients in group C1 probably has not the same origin of the disease. All these patients have Sjögren, have not the same origin that these patients in group C3, and the same is group C4. And you can see, for example, if you look at the secretion of different types of cytokines, like different type of interferon, you can see they are different depending on the groups. So just to show you that the future in autoimmunity is to better stratifying the patients, because probably the treatment which will work will not be the same in C1, C2, T3, and C4. The second part of the talk would be really to show you that now we have promising drugs. And that's very important for you, and to show you that it's very important really to participate to clinical trials, because now it's a time where we have clinical trials in this disease. And I show you just a flavor with 3 recent publications. The first one is a concept which is really easily understandable. We have numbers of drugs in autoimmune disease, and these drugs are all drugs that are used in rheumatoid arthritis, in systemic lupus. And the idea of this group from Netherlands was to combine 2 classical drugs, very well known, hydroxychloroquine and leflunomide, because one of these drugs targets the B cells and the other one is going to target the T cells. So because the patients are heterogeneous, it makes sense to combine different drugs. And we do know that these drugs are perfectly safe. So you see on this scheme that one of the drugs is going to target mainly the T lymphocyte, and this other one to target mainly the B lymphocytes. And they made a clinical trial, a very simple clinical trial with only 29 patients, and they could evaluate the effect of the combination. You can see here the combination in this dark square here. And you know what we do a clinical trial, we have half of the patient treated with placebo and half of the patient treated with the active drugs, and nobody knows. Neither the patients nor the clinician knows what the patient receives. And that's a way of evaluating the response by the SDI. SDI is a score which is going to evaluate the systemic complication of the disease. And you can see that clearly there is a difference at the 24 weeks between the combination of these classical drugs and the placebo. And they also look at the symptoms of the patients. And you know that the symptoms is well evaluated by this score, which is called ESPRI. And ESPRI, it is a visual analogic scale of 3 types of symptoms. The main symptom you have every day, which are dryness, pain, and fatigue. And you see there is a decrease with the drug and no decrease with the placebo. And in these trials that also found that you see when you look at specific domains like fatigue and pain, there is a difference. And also the salivary flow, the unstimulated salivary flow increased, you can see, with the combination of drug and doesn't change with the placebo. So it's encouraging. It's a very small study, but it is a combination of classical drugs. We may have these drugs in every country. A second example of a new positive clinical trial is to target now what we call the B lymphocytes, because in this disease, there is really an overactivation of this subset. And this drug is developed by Novartis. It is iscalimab. Iscalimab is going to inhibit a protein, which is called CD40, and which is going to play a role in the activation of the B cells. And again, they did a first small clinical trial recently published. You can see all these studies have been very recently published. Again, the drug active and the placebo during 3 weeks. And you see after 3 weeks, everyone receives the drug. And that's interesting because when we have this type of clinical trial, it is much easier for you, patients, to accept to participate because you are sure that at least at one time you will receive the drug which is tested. And I think that this design should be encouraged vis-a-vis pharmaceutical industries, because of course it is much easier for the patients to accept to participate to this type of trial. So what is the result of this trial? Again, we look at the SDI. SDI, it is this marker of systemic activity. You see here that there is also a difference at 3 months between the active drug and the placebo. And when the patients who were on placebo received the active drug, you see in the second part of the trial, there is a decrease, which is not as spectacular as the initial phase, but there is a decrease when the patients receive the active drug. And that's something which is also very encouraging. And now there is a second trial, a larger trial, which is called phase 2B with this drug, which is ongoing. And they also looked at different other outcomes. That's the SDI we have seen. But you see, this drug, it's not significant because when you see when 0 is reached, it means that it is not statistically significant. But when it is on the right, it favors an activity of the drug. When it is on the left, it would favor an activity of the placebo. And you see that everything is on the right. Even if it's not significant, the symptoms of the patients, the fatigue, the appreciation of the disease by the physician global activity, the appreciation of the disease by the patient global activity, and SF36 is a measure of quality of life. So it is an encouraging drug, and we are waiting now for the results of the very recent trial, which involves more patients. And the last example is not yet published, but I would like to share with you this study because it is also very encouraging. It is a combination of 2 drugs which exist, which also targets the B lymphocytes. The rituximab is given sometimes in your disease. Maybe some of you have received rituximab because it may work in some complications, but the big trials with rituximab were not successful with rituximab alone. So the idea is to combine rituximab with another drug, which is belimumab, which also targets the B cells. And belimumab is approved in the treatment of systemic lupus. And there is a rationale for combining both drugs, which could work together better than one drug alone. And so GSK designed this clinical trial. And you see there are 4 groups. there are 4 groups. One group with, as you can see, belimumab alone, one group with rituximab alone and belimumab placebo, and one group with a combination of the 2 drugs. Now, this group has both placebo and this group has belimumab alone. So it's a little complicated, the design, but you need absolutely that to know what the combination of drug does. One drug alone and a double placebo. And look at the results. It is the percentage of patients who responded to the drug. If you look at week 68, so it is 1 week, 1 year and a half, almost. In green, the placebo. In purple, in pink, the combination of 2 drugs, and in blue and orange, only 1 drug. Really, it is encouraging to see that the combination of both drugs seems efficient compared to placebo. And what was very important, the safety of the combination was very good. So it is something which is also very promising. And when we look at another outcome, which is the salivary flow, you see again that it doesn't change with the placebo, with 1 drug alone, but when you have the combination, there is an increase of the salivary flow over time. And the last example, the 4th example of a successful trial, has been very recently published in The Lancet. The Lancet is a very famous journal. It is one of the most famous medical journals. You have the 2 most famous are New England Journal of Medicine and The Lancet. And this drug also is going to target the B lymphocytes. And it is an anti-BAFF receptor antibody. It's called ianalumab. It is also developed by Novartis. And so it is just to see if there is a dose effect, 3 doses of the drug and the placebo. And what you can see clearly, again, you have the decrease of the SDI. You have the placebo here in dash, and the 3 doses, as you can see, low dose, middle dose, and higher dose. And clearly, when you have a high dose, you have clearly a decrease of SDI, which is different than the decrease observed with placebo. So there is a dose effect. And so this drug really can be interesting in this disease. You can see here the dose effect and the percentage of responder patients with the high dose compared with the placebo with this definition of response. So again, this drug is very promising. And now Novartis is beginning a phase 3 trial. Phase 3, it means that there are a lot of patients all over the world which are going to be included in this new trial with ianalumab, this anti-BAFF receptor antibody. So that the 4 examples, combination of classical drugs, association of 2 biologics, rituximab and belimumab, or new drugs targeting the B cells. So that's really where we are now in the clinical trials. And in this table, I showed you the results of very recent clinical trials. I presented the 4 green ones, which were positive. Of course, some of them are negative. And you can see on this slide that some of the trials which have been conducted are negative. But it is the first time now for 2 years that we have the first positive. It is phase 2 trials with a few patients. So we have to confirm with phase 3 because before this drug could be available for every patient. And what we can see, and we recently with Raphael Serror and Gaetan Nocturne, my collaborators, we made this paper just to show the different targets, the different type of drugs, which could be efficient in this disease. And you can see a lot of them could be efficient. So now we have the targets. We have made progress on pathophysiology. We may test some drugs which really may be specific of a target. And of course, the dream is to have a stratification of the patient, because maybe patient A will respond to anti-BAFF receptor, patient B to CD40, patient C to JAK inhibitors, for example, depending on the biomarkers. And the last part of the talk, I'd like to speak of you of the NeceSSity project. What is a NeceSSity? NeceSSity means new clinical endpoints in primary Sjögren syndrome and interventional trial based on stratifying the patients. So you find again the same concept: stratification, biomarkers, clinical outcome to be used in clinical trials. So it is a project which have been accepted by the European Commission. It is what it calls an IMI, Innovative Medicine Initiative. And it is an original project because it is funded by the European Commission, but also by a consortium of pharma industries. So we have to work together, academics, industrials, and patients. So there are two leaders. One is from the industry, Peter Gergely, and one is academic. It is myself, so I coordinate this project. And what is the vision of the project? We do know that the burden of Sjögren's is high for the patients and for the society also. We do know that we don't have yet any drug which has clearly demonstrated in a phase 3 study that it decreases the symptoms and improves the evolution of the patient. And why? We have drugs in rheumatoid arthritis. We begin to have drugs in lupus. We don't have drugs in Sjögren. Why? Maybe we thought that the primary endpoints we use in our clinical trials maybe have limitations. You have seen that the SDI only measures activity of the disease, the systemic activity, the systemic complication. ESPRI only measures the symptoms. So it is two facets of the disease, but there are also all facets, other facets. There are the tears, the saliva. And so the idea was to develop a new score which could encompass all the domains of the disease. And the second objective, the second vision, is to say that maybe we have difficulties to find a treatment which works because the disease is heterogeneous. So we have to find new biomarker for stratification. And it is also one of the objectives of NeceSSity. So we are 25 in NeceSSity, as you can see. We are 20 academic partners all over Europe, as you can see, in the different European countries. We have 4 companies on board, and they don't give some money by cash, but they give some data from their clinical trials. So it is the way of participating: Novartis, GSK, BMS, and Servier. And very importantly, we have the patients. And so the patients are represented by the French Patient Association, AFGS, but it is just for representing all the European Patient Association, because at the time of the application, we did not have yet Sjögren in Europe. So AFGS is just representative of Sjögren Europe. And we have 3 objectives. The first one is to find new biomarkers for stratification. The second one is, as I said, to develop a new clinical outcome, a new score, which should be used in future clinical trials for assessing all the aspects of the disease. And the third objective is to set up a clinical trial for validating this score, because it's the only way of validating the score is to do a prospective clinical trial. So how are we organized? There are different work packages, as you can see here. And among these 10 work packages, all the stakeholders participate. And the patients participate actively to all these work packages. Maybe less actively in the biomarker work packages, the 2 ones, but in the clinical trial, in the clinical endpoints, in the ethic, management, coordination, dissemination, communication, the Patients Association participates very actively. So the budget of the project is 15 million. Half is given by EU and half by the companies. By the company is not cash, as I told you. It is availability of data from the clinical trial, and it is a 5-year project. a 6-year project, sorry. So where are we now? We have begun now 3 years ago, and we are really in good advancement of this project. In the biomarkers, it's always complicated to work on biomarkers with different groups because we have to share the samples from one center to another, from one country to another, from industry to academic, from academic to industry, and you imagine all the requirements now it requires. All the documents, all the authorization. And we had the COVID-19 among that, above that. But now we could share some samples. You need between 9 academic and 1 industry samples, data sets, and really we make really good progress for stratifying the patients and to find new biomarkers. The second objective is to develop this new outcome for clinical trials, and we call that the STAR. STAR, I see it's not written on the slide what it means. Sjögren syndrome tool for assessing response to treatment. That's a nice name, STAR. So it's a tool to be used in clinical trials. So you see, we had a very important patients' contributions. 20 patients participated to the development of STAR. We also had the participation and we worked with collaboration with the EMA, European Medical Agency. And we also work, now we began collaboration with FDA and with OMERACT, which is an international organization for developing scores of evaluation for evaluating rheumatic disease. So it is the justification of the STAR, needing of stratification, evaluation of the disease with SDI and S3, which are limited, and the absence of inclusion of outcomes measuring the glandular function. So what we did is we firstly analyzed 9 previous clinical trials. And we looked in these trials, what are the domains which are important for the patients and what the items for measuring each domain. And we organized a Delphi survey. And you see in this Delphi survey, we had 79 experts. So it's really a collaborative work from different continents, as you can see here. And we also had 20 patients, also from different continents. You see a lot of patients from the US participated to this Delphi. And we identified the domains to include. And after we constructed STAR, because there was different ways of construction, and we evaluated the sensitivity, specificity in the previous clinical trial. And now we are going to begin the final step, which is the validation in the prospective clinical trial. And I'm very pleased to tell you that all this process has been now published 3 weeks ago. It's very recent in the Annals of Rheumatic Disease, which is the top 1 journal of rheumatology all over the world. And so we have the final version of the score. And you see, we have 5 domains. Systemic activity, we measured with SDI, the clean SDI. The patient-reported outcome, we measure it by S3. We have the third domain, which is lacrimal gland function, measured by the Schirmer test or a more complex test, which is ocular staining score. The salivary gland function, which is measured by the unstimulated salivary flow or by ultrasound. We incorporated ultrasound in the score. And we have a biological domain, which is measured by the total level of immunoglobulin or by the level of rheumatoid factor, because we do know that it is a marker which could be prognosis of occurrence of lymphoma. And for being responder, we need 5 points. And you see the number of points, the weight for each domain. So for being responder, it's mandatory to have response of one of the major domains, which is the systemic activity and the PRO. And you see all the steps of the validation, construction and validation of the candidate stem. So it was a huge work, and I'm very proud that now we have achieved really this objective. Now we have to validate this score. And to do it, it is the third objective of NeceSSity. To do it, we have to run our own clinical trial. And it is really something which is very hard work for organizing that in 8 countries, 35 centers, and we are going to include 300 patients in this clinical trial. And you see, because we did not have available the new drugs which are in development that I presented you previously, we chose to confirm the first data from the Dutch study with a combination of leflunomide and hydroxychloroquine. And we also included a third group with a combination also of hydroxychloroquine and MMF, which is a drug which is frequently used in lupus. So you have one arm with placebo and one arm with two arms with each of the combination. And you can see that another originality of this trial is that because we use drugs which are very well known and which are safe, we also included patients in this cohort 1 without systemic activity, which is measured by SDI. SDI is low, but the symptoms measured by ESPRI are elevated. And it's important to see if this drug may work in also these groups of patients. And I'm very pleased to announce to you that we have included the first patient in this clinical trial yesterday in my center in France. So we are beginning now this project in 8 countries, as I told you, and the objective is to include 300 patients. And of course, we will need you for advertising this trial in the different countries. In this trial, we will also test new non-invasive assessments, because when you measure efficacy of the disease, sometimes we ask you maybe to do again a biopsy, lip biopsy, and of course the patient doesn't like very much. And so it's important to measure with new tools how a drug may work. And for example, we have this application which is called PEPS. PEPS is a web application on your mobile phone, just in order that you can measure pain, dryness, and fatigue every day. Because when you participate to a clinical trial, we ask you to measure dryness, pain, and fatigue at baseline, at 3 months, 6 months. But what happens between? And you do know better than I do that it is very variable along the day. So this application is very easy to use. We tested it in my centers, and patients like it, and we will measure so every day to see if there are variations. There is also something which will be tested in Birmingham, UK, which is a biosensor. It's just a watch and a patch on the breast to measure different data, the heart rate, the number of feet, and to try to evaluate also the fatigue. Ultrasound will be used for measuring the effect of the drug on parotid. And new non-invasive ophthalmologic procedures also will be tested in this protocol. So you see, the idea is really to also validate a new way of measuring the disease by non-invasive procedures. I think that's it. So I thank you very much for your attention. And here's the entry of my hospital during winter. Today it's very sunny in France. And I will be very happy to discuss with you and to respond to your question. Thank you very much.
Hi everyone, my name is Anna. I'm also a member of Sjögren report, and I will be handling the Q&A. Please have your questions written on the Q&A dedicated space. Professor Xavier, Mariette, I would like to thank you so much for this very comprehensive, and I would dare to say overwhelming presentation. It gives us a lot of hope. So I would start with one of the questions. Are there any novelties in terms of diagnosis
Thank you for,
lately?
thank you for this question. So the diagnosis of Sjögren is mainly based firstly on the symptoms, dryness, fatigue, and pain, but also can be done in front of a complication of the disease. For example, purpura, bronchial involvement, neurological involvement, parotid swelling. So all these symptoms may reveal the disease. For the diagnosis, the first exam to do is to look at autoantibodies and to look at anti-nuclear antibodies and anti-SSA, which are called also Ro60 antibodies. 2/3 of the patients will get antibodies. And in these patients, the diagnosis is almost done. But 1/3 of the patients do not have these specific antibodies. For doing the diagnosis, it's mandatory to do the lip biopsy, the minor salivary gland biopsy. It is invasive, but when it is done in a reference center, it's really something which is easily done, very easily done. And on the biopsy, you see this infiltration of lymphocytes within the gland. So now there are some research for looking if we could replace the biopsy by ultrasound. And ultrasound could be efficient. They cannot replace completely the biopsy. But for example, we have demonstrated that when you don't have any anti-SSA antibody, and when you have normal ultrasound, the probability of a Sjögren's is very, very low, and you can almost eliminate the disease. So ultrasound could be one new tool for helping the clinician to make the diagnosis of the disease.
That is great news to have a non-invasive
Yeah.
tool to check on the salivary glands. Another question here is, how do you sign up to be part of the NeceSSity trial?
Okay, so I will give you the list of the centers. As I told you, we have 8 countries and 35 centers. The countries are Greece, Italy, Spain, France, UK, Netherlands, Sweden, and Norway. And we have different centers in these countries. So the process is to contact one clinicians in one of these centers to see if you requires the inclusion criteria. You do know that in every clinical trial there are inclusion criteria, but in NeceSSity they are easy to achieve because we want to include all the type of patients with Sjögren, not only the patients with systemic complications, but also the patients who suffer from dryness, fatigue, and pain.
That's really nice. That's really nice. That's also a novelty. It's
Yeah.
a new avenue in clinical trials.
Absolutely.
Another question here is, can PEPs be made available to the Sjögren's community long term to do longitudinal research?
Absolutely, that's a very good question. So PEPS has been built in the context of NeceSSity, and the patient association is a partner of NeceSSity. Thus, I think it should be possible, and we have to discuss probably with Coralie to see how PEPs could be available to the patients, to the Sjögren Europe Association. So PEPs has been developed by one partner of NeceSSity, who is DV Cornec from Brest. PEPs will be used in the clinical trial. I don't know, Coralie, if you had some preliminary discussion with DV about the possibility. Yes, I leave you respond to the question.
Yeah, we did, absolutely. It was the last General Assembly. I obviously tested the web app during the trial, and I obviously thought of all patients, obviously for research, but for day-to-day routine and, living with the disease. And as you said, just to evaluate how patients are before going to see the clinician. So yeah, we already talk with DV about this app, and we'll probably see later what are the possibilities to work together and to offer this opportunity to the Sjögren community. Yeah, absolutely.
Thank you, Coralie. Another question, a challenging one, I would say. So there is no German center involved in NeceSSity. So is there a possibility to participate as a German patient? This is a German patient asking.
Yeah, I see the question, Christian. Yes, firstly, I apologize for not having German centers on board. It is just, it's not a willingness. You do know that how the friendship between France and Germany is important and is something so important in Europe. And no, it's just, how can I tell that? It's a huge work for finalizing a project like NeceSSity. And we had on board Thomas Dorner, which is a friend of mine from Charité in Berlin. And Thomas wanted to participate, absolutely. And finally, I tell you everything. Charité didn't want to participate because the amount of money that Charité could obtain from NeceSSity was not enough. So it's just a question of money. I apologize. So no, you will not be able to participate and to find a German center.
Thank you, Professor Xavier. Mariette, another question regarding the centers involved in this trial. Is there a timeline for the different centers, or will they start all at the same time? Which countries will start first or after, or they will start recruiting all at the same time?
So the objective is to start at the same time, but it takes some time for finalizing all the contracts and all the logistics. So until now, we have opened 6 centers in France. We are going to open the Swedish centers very soon, probably an Italian centers in Napoli, and the Greek centers, it is almost ready, and they will be open in the next week. So the objective is to open as soon as possible all the centers all over Europe. And the duration of inclusion will be 2 years. So we have planned 2 years for inclusion. And 1 patient will be followed for 9 months in this clinical trial.
Okay. Another question is, what do you know about Sjögren syndrome and interstitial cystitis? Can some of the medication you tell us about help for this complication?
Yeah, that's a challenge because interstitial cystitis is a symptom which is very difficult to improve, even when it is not associated with Sjögren. It can be primary also. The mechanism is not so clear. It could be linked to an infiltration of the epithelium by the lymphocytes also. I must say we don't have any symptomatic treatments yet, and we don't know if these drugs, is the immunosuppressive drug, could work or not work. So it has to be assessed.
Thank you. Another question is, how is neuro-Sjögren's taken into consideration?
Actually, neuro-Sjögren is very heterogeneous also. Among the heterogeneity of the disease, the neurological manifestations are also heterogeneous because they can be very different. Sometimes it is sensory neuropathy, so pure sensory, pain, itches, dizziness. Sometimes it can be motor involvement. And when you are a motor involvement, so a muscle which doesn't work because of involvement of the nerves, in most of the cases is due to vasculitis. And the treatments are not the same depending on the type of neurological symptoms. We think, just to summarize, that when the neuropathy is purely sensitive, it's difficult. We don't have any good treatment which can treat the cause, and we use symptomatic treatments, drugs which are going to decrease pain. When the disease involves also the motor nerves, immunosuppressive really may work. Sometimes when it is linked to vasculitis, we may use steroids, we may use rituximab. So it depends on the type of neurological involvement.
Okay. Can there be a consensus between countries to standardize diagnostic criteria and pool research from all countries?
Yes, I think we have achieved this goal because now we have consensus EULAR and ACR criteria. And you know that I have participated actively to the establishment of these criteria. So they are now recognized in the US, in Europe, in Asia. Some Asian countries continue to have their own criteria, but they are very close. So I think now the criteria for diagnosis of the disease are consensual all over the countries.
That's good to know. Now a challenging question: what can be done to improve the detection of potential rheumatologists and train these doctors in the field of rare diseases and in particular in Sjögren's disease?
Yeah, that's a challenge because numbers of GP don't know the disease very well. Numbers of specialists also, finally, even rheumatologists sometimes don't know this disease very well. So there is an effort which is organized by the European Commission with the European Research Network on rare diseases. And we have an ERN, and I do know that you participate, Anna,
Yes,
and other
and Coralie as well.
Yeah, Coralie in this ERN. And this ERN is called Reconnect for rare connective tissue disease. And in this network, we have lupus, we have scleroderma, we have myositis, and we have Sjögren's. And I am coordinating these actions on Sjögren's. And in this, so we have reference centers in different countries. And each reference center in each country has to work for teaching other centers, other rheumatologists, for having in each country a network of specialists which can really be called reference center for Sjögren in all the European countries.
Thank you. We hope this network really grows, and I would like to add that recently we've had new members joining the network, so I hope that soon all these actions on Sjögren can grow and can become more and more promising and to shine light on the disease in particular. So another question here is how to make a diagnosis when the clinical criteria are important and consistent with this disease, but remain timid or discordant with the officially recognized biological criteria. What is the medical attitude to have?
Okay, that's a great question also. So the classification criteria are criteria for classification, as it is really indicated. So for doing clinical studies with some patients which are roughly the same. And as I told you, even with this criteria, there is a big heterogeneity between patients. But these criteria sometimes are not useful for the diagnosis. And maybe, for example, you may have a diagnosis of Sjögren if you have dryness, mouth dryness, eye dryness. You don't have antibody and you have a lip biopsy with not the focus score which is required, but we have infiltration. So because maybe the biopsy, if you had made the biopsy in another part, you would have the focus score which is required. So at the end of the day, it is the clinician who makes the diagnosis. The criteria help, but the clinician makes the diagnosis. But what is also very important to understand to you patients is that the three main symptoms of the disease, dryness, fatigue, and pain, may occur in numbers of other circumstances which are not Sjögren and which are not an autoimmune disease. You may have pain, dryness, and fatigue with drugs, for example, dryness when you use some antidepressive drugs, when you use morphine, analgesic drugs, you may have dryness. And sometimes in these symptoms that we don't understand very well, we rheumatologists, we call fibromyalgia, pain, fatigue. And in fibromyalgia, you may have dryness, even though you don't have any drugs. And until now, we don't know the origin of fibromyalgia, but we don't have any arguments to say that it is an autoimmune disease. So it would be a pity to treat a patient called Sjögren who have only just fibromyalgia with an immunosuppressive drug. So these symptoms may be very disturbing for the patients and not being due to Sjögren. So in your association, it's the reason why we had a lot of discussion with the French association now 20 years ago. And I advise the associations to change the name and to say Association of Sjögren and Zika syndrome, because you may have a Zika syndrome without any Sjögren because of other causes, you know, and it's important. So, and numbers of patients, they say they think they have Sjögren, but actually they have no antibody, normal biopsy. So they have the symptoms, of course, because they say they have the symptoms. So it's true, but it is another reason for that. And we have to work more for understanding.
Differential diagnosis is really important. It is so important to know what you have when you have Sjögren's, but it is also extremely important to know that you don't have Sjögren's. So that's why a differential diagnosis is very important. Another interesting question that among the patient community is always raises several discussions. Is Sjögren's really a rare disease, or is it just rarely diagnosed? What is the prevalence in Europe? Nobody has ever been able to answer that, or better, we get answers from different countries, different answers.
Yeah. One of the discrepancies is linked to what I said previously. In some, for example, the US Association in this booklet said 2 to 4% of Americans suffer from Sjögren. I think it's not right, but at least 2 to 4% of Americans suffer from dryness, and probably 5 to 10%. Okay, so you may have dryness without having Sjögren. When you speak of Sjögren with the criteria we spoke about, it's criteria which actually demonstrate that it is an autoimmune disease, either autoantibodies or lymphoid infiltrates on salivary glands. The disease can be considered as rare. What is the definition of rare? The definition is 1 over 2,000 patients. Personally, I think it is a little above this threshold. And the prevalence should be around 0.1%, 1 over 1,000. So after the game, if it is a rare disease, you may have access to funding for research, for drug development, for rare disease. And it can be easier to develop a drug if it is a rare disease. So it's the reason why you see these discrepancies also, because sometimes you may have interest to call the disease rare disease for getting funding for rare disease. And sometimes when you want also to emphasize that it is a health problem, you can have interest to say it is a frequent disease, a common disease. The truth is between. But among the autoimmune disease, the systemic autoimmune disease is not so rare, because the most frequent systemic autoimmune disease is rheumatoid arthritis. Rheumatoid arthritis involves 0.5% of the general population, 0.3 to 0.5. If Sjögren is 0.1, you see, probably it is the second most common systemic autoimmune disease. It is more common than lupus, or myositis, or scleroderma. And another reason for that is you can also focus only on primary Sjögren. If you include Sjögren associated with other connective tissue disease, Sjögren associated to RA, to lupus, to scleroderma, it is more common. So it is between the two.
Okay, thank you. Another question is, as you know, all Sjögren's patients use and need to buy for their everyday living comfort, topical gels for the eyes, saliva replacements, and they are mainly not reimbursed. And patients often believe that this is because their countries, their national health systems do not recognize the importance of these comfort pharmaceutical products, let's call them like that, for these patients. How do you think this can be facilitated, the recognition of these pharma products to give quality of life and comfort to Sjögren's patients?
Yeah, that's a very important topic. Firstly, you do know that in every country, all the health authorities, all the political powers are very reluctant to reimburse new drugs because it is always money which is spent in addition to money which is already spent for health. I think for helping authorities to reimburse, we have to demonstrate that it is a disease which is perfectly defined and the criteria are important. It's the reason why it is important also to change syndrome for disease, just to show the politician that it is a disease clearly defined. Secondly, we have outcomes which are clearly defined, and it's the reason why that I think that STAR will be very important. So we have an outcome. Outcome can be used for clinical trials, and the clinical trials and this outcome involves different domains, and among these domains, the patient-reported outcome are very important. And I think it is just what you are doing in your association, what we are doing as clinicians and researchers, that progressively the authorities will accept the fact that these drugs are not only comfort drugs, but are very important for quality of life for the patients.
Thank you so much. It's been a very clear and interesting approach, the one you just gave us. A final question here from a patient that says that she's intolerant to all medications she has tried so far. And because of side effects, she gets several side effects from different medications, but no one has been able to answer this for her. None of the doctors she's seen so far, she's seen several doctors, tried several different medications, but she always develops side effects and needs to stop the medications. Is this common in children's?
No, it's not common, but every case is, of course, is specific. So it's difficult to respond specifically to this patient, but it's not a common description. Patients with Sjögren are not particularly susceptible to side effects to the drugs. So it's specific to this patient, and she has to speak with her clinician to see what can be done for them.
Okay, thank you so much. I have no further questions. I don't know in the chat, I think we've gone through all. There's a comment here. Thank you for the nice and informative presentation and your great effort for patients. Patients are getting more and more informed, sometimes better than us physicians.
That's true.
But there's a question here in the chat, not on the Q&A, just asking if COVID-19 can worsen the symptoms of the disease. If you have any information on this, Professor Xavier, Mariette.
Yeah, that's a good question. We have conducted, maybe you're aware of that, a big registry for assessing the safety of the vaccine. And overall, the vaccine is safe. So if you look at, we had 5,000 patients all over Europe with autoimmune diseases, RA, lupus, Sjögren, and we saw a flare of the disease in 5% of the cases only. It could be just linked to hazard. So globally, the vaccine is safe. The infection, I've seen 2 or 3 patients with flare of the disease after COVID infection, and that's not a surprise, because we do know that autoimmune diseases can be influenced by any infection. When you get an infection, you increase some of the cytokines which may play a role in autoimmunity, like type 1 interferon, for example. And so it is something which can be described, but we know that with other type of infection, with flu, with pulmonary infection. So yes, it's possible that a COVID infection could lead to a flare of disease. It's not common, but it can occur.
Okay, thank you so much. I think there are no further questions. Thank you for this great time together. I hope all of you in the audience enjoyed it as much as I did. Professor Xavier, Mariette, thank you again for your availability, for your time, for your interest in cooperating with us and contributing to patients' health literacy and to improve their information on the disease. Coralie, would you like to say something? The floor is yours.
Yes, said everything, Anna. Thank you again, Professor Mariette. Thank you, everyone from all countries all over the world for your attendance. We really hope you enjoyed. Other webinar will come soon, so you can find information on the Sjögren Europe website and on our social media. Thank you, Anna, for your excellent question and answer session. And a special thank you to Cathy, our board member, who organizes all those webinars in a masterly way. So have a good night, everyone. Take care of yourself and see you soon. Bye-bye.
Bye-bye.
Sorry,
Thank you
guys,
so
there is
much.
another
Bye-bye.
question.
Oh,
Sorry.
is it possible that Sjögren has something to do with amalgams that include mercury?
Yeah, it has been something which has been suspected in the past. No, I will say no. There was some studies on that, and sometimes mercury cannot be tolerated. It's not used anymore in amalgams now. But I've seen, for example, some patients having removed all their amalgams because they thought it was the cause of the disease and it didn't change anything. They had to spend a lot of money for doing them again. So the response is no.
Thank you so much. I think now we're done.
Okay. Thank you to you.
Thank you.
Goodbye, everyone.
Good luck
Thank
with
you
your
so
report.
much.